Okay. We will discuss today the structure, the monomers. It is the building block of the DNA. It is called the nucleotides. We will today know the nucleotides. We will go through the structure of the nucleotides, and you will get to build the DNA using simulation. Function of the DNA is the maintaining and storing the genetic information of all living things, human, animal, and plants. And also it will make the protein synthesis, determine the protein synthesis, which we will go through today. Okay. When I ask the students, do you know the DNA? They say, yes, yes, of course, we know DNA. DNA, it is genetic information, it is the genes, it determines the characters of the living organism, and they know very well the DNA. But when I ask the student, do you know what is RNA? Most of them, they don't know. They don't know what is the difference between DNA and RNA and what is the function of the RNA. Actually, the RNA, it is the most working or the hard worker nucleic acid in the living organism. It is more than the DNA, but the DNA is more popular than the RNA. The DNA is just sitting in the nucleus, containing all the genetic material, and that's it, doing no work. But the RNA today we will see how it is very active, it is doing all the work, and it is a very important part of the protein synthesis, which is a very important process for the living organism. When we look at the two, DNA and RNA, can you tell me what is the difference between them? We have two main differences here. Want to use the chat to answer? Think about it and have 10 seconds to tell me what is the difference between them in DNA and RNA. So DNA is double-stranded, very good, but the RNA is single. One more difference is that if we look here, the nucleotides or the nitrogen base. I have here cysteine, guanine, adenine on both sides, but here in the RNA I have uracil, and the DNA I have thymine. It is very important and crucial difference between them. We will use this in the DNA replication and protein synthesis. We need to know the difference. And in base pairing, we have two types of nitrogen base. We have the purines and pyrimidines. The purines and the pyrimidines are nitrogen base in the nucleotide. Okay, miss, what is the nucleotide? The nucleotide, it is the monomer or the building block of the DNA. The smallest thing of the DNA. It's called nucleotide. The nucleotide, as you see here, it is like blocks. It is only made up of 3 parts. Phosphate group, deoxyribose sugar made up of 5 carbon atoms, and nitrogen base. Nitrogen base is made up of 4 different kinds. A, G, T, and C. As I told you, adenine, guanine, thymine, and cysteine. Okay, miss, but we saw before uracil. Yes, uracil in the RNA. Here we are not talking about RNA. I'm talking now on the structure of the DNA. Then we will know the RNA, what is the function in the protein synthesis. Okay, we will use a simulation to build the nucleotide and to build the DNA double strand. Okay? To help you to understand the difference, to help you to build the DNA and know how the bonding between the base pair, what are the main bonds between them, how they are pairing. This is very important. Building block again is the nucleotide. So we are going to build the nucleotides. We have repeating parts common in all nucleotides, which is the sugar, pentose sugar, and the phosphate group. This will be linked to one of the nitrogen base. So that's why I have 4 nitrogen base. That's what is giving the variation of the DNA, the nitrogen base. So to build another one, again, I use sugar, phosphate group, and another nitrogen base. They are going together and base pairing in an alternative way. So we have a very different and various types of DNA and a lot of variations in the living organisms because of the nitrogen base. Let's start. This is the fun part to build the DNA. I have one strand. I need to build the complementary strand. Again, he's reminding you that A pairs with T and G pairs with C. To build T, I need to pair it with only one nitrogen base, which will pair with like a lock and a key, key and lock. T is always pairing with A. Okay, let's go to the next. C. C will always pair with G. Hydrogen bond in the middle. And now we build a double strand DNA. So now we know the pairing of the DNA of the nitrogen base, how we build the double helix, what is the structure of each nucleotide, and everything is good. We can move now to the protein synthesis and DNA replication. Again, this is a misconception. Most of the students, they don't know the difference between replication and transcription. Replication is only I'm doing this in cell division process. If I am making DNA from DNA, I'm making another copy of the DNA in the growth of the living organism, in the growing baby. If you have a wound, you need to renew your skin. This is happening by the mitosis cell division. So DNA is replicating, having the same copy of the DNA. And I have the transcription. In the transcription, it is happening in protein synthesis, in making protein. The DNA, it's making RNA. Here we will talk about the RNA and the function of the RNA, how it is the active one. It's doing all the job in the process of protein synthesis. Another thing we have in the protein synthesis, we have making protein from the RNA. So we will know how to make RNA from DNA and how to make protein from RNA in translation. If I'm doing mitosis, cell division, I want to make replication, making DNA from another DNA, we will base pair the nitrogen base here and the nucleotide. So we will here start pairing them. G always pair with what? Always pair with C with G, T with A, T with A, C with G, C with G, and as you see, it is any order. As long as you're pairing G with C, A with T, and so on. G, T, T. So now I make another strand, another complementary of the same DNA. This is called replication. I am making another DNA. Remember, we said that A paired with T and G with C in the DNA. In RNA, if I want to make RNA, there is no T, there is U, uracil. So G with C in the RNA, A with U. You cannot write T in the RNA. Like here, I am making transcription in protein synthesis. Can I write T and write A? Yes, because we have A in the RNA. But here, can I write A and the complementary T? No, you cannot write T when you are making RNA. This is RNA. This is DNA. So it's okay to have A and T. But when you make the new RNA complementary strand, and in a minute we will know why we need to make RNA from DNA. A will pair with what? With T? No, I cannot write T ever. Here in the RNA. I will write U. C with G? Yes, no problem. C with G? Yes, no problem. G, C, C, T, A? Yes, no problem. T, A, and the last, you cannot write T here. You need to write uracil. Okay. Why not? Why we need to make RNA from the DNA? This is happening in the protein synthesis process, in making protein. All living organisms need protein. So, which organelle in the cell making protein? The ribosome. Okay, Why we are doing this? Why we are making RNA? Why we don't just make DNA inside the nucleus and that's it. This DNA go outside to the ribosome, center of protein synthesis, and make the protein. Why we need to make a copy of the RNA, of the DNA on the form of RNA? Can the DNA leave the nucleus and go outside to the cytoplasm and do all the work? Anyone wants to answer? Can we do this? No, we cannot. The DNA cannot leave the nucleus. Why? Because it is very big, very, very big. It's folded inside the nucleus. It is very big. Second, we cannot the DNA leave the nucleus. It is like the safe nucleus keeping the genetic material inside the nucleus. It cannot leave the nucleus. Can be destroyed, denaturated, or can be broken outside the nucleus. We need it to be safe in the nucleus. So we need to make a copy of all the genes, all the genetic material in the RNA. And this RNA, messenger RNA, will take all the information and go to the ribosome outside in the cytoplasm to make the protein. Take the order from the DNA with all the information, all the data, all the recipe, and go to the ribosome to make the protein. Okay. We have three types of RNA. RNA is very important, and we have three types of it. We have messenger RNA, mRNA, ribosomal RNA, rRNA, and transfer RNA, tRNA. We usually use this analogy to know the difference between the types of RNA. We said that the nucleus, it's like a safe, keeping the cookbook, the main book. It's very important, the DNA inside the nucleus. So to make a copy, to make the protein with all the data and information on the DNA on the cookbook, I need to make a copy. So the messenger RNA, the one we will build from the main DNA, it is like a photocopy machine. It is taking all the information from the DNA and it will send it outside the nucleus to the ribosome, to the ribosomal RNA inside the ribosome. This is the cook. It will make the recipe. It will cook everything together. To cook ribosomal RNA, it needs material, it needs ingredients. Who will bring the ingredients? Yes, messenger RNA, yes, okay. And we need another ingredient to make the protein. The protein is made up of amino acids. So the amino acids is the ingredient. Who will bring them to the ribosomal RNA? Transfer RNA. So messenger RNA, they make a copy of the DNA. Ribosomal is like the cook, it cooks everything. And the tRNA bring the amino acid and give it to the ribosomal RNA to make the ice cream or the protein. So inside the nucleus, happening transcription, DNA make a copy called RNA or messenger RNA. And this messenger RNA leave the nucleus and go to the cytoplasm, to the ribosome, the site of protein synthesis. And who will bring the amino acid, the ingredients? tRNA or transfer RNA. Will open inside the nucleus, base pairing as we said, A with U, C with G, and so on. I am making new RNA, so as you see, we have U. A pairing with U, not pairing with T. And so on. So make a complementary strand. To make a complementary strand here, we have, again, I am making messenger RNA, again, remember T with what? T with A with T, no, A with U, C with G, G with C, C. I can write T in the RNA, no, I'm making messenger RNA, remember, I will put U, T with A, G, T with A. Okay, this is, I am making now messenger RNA. What will happen next? This messenger RNA will leave the nucleus and go to the cytoplasm, to the ribosome, to ribosomal RNA, bring the amino acids, and to make the translation, to translate the messenger RNA to protein. What is the building block of the protein? Amino acids. We need to do decoding, translating the RNA, the nucleic base, into the sequence, into amino acid sequence to make a long chain polypeptide protein. First thing, I need DNA in the nucleus. We will make the mRNA base pairing as we said. Then I need to translate each three into one amino acid. I will use this. Table here to translate the codon and put the right anticodon. So you have I have AUG. I need to look here at AUG. AUG is here. We always start with AUG. It's called methionine. It is the starting amino acid. Should start the process. Then what comes next? CGU. You need to go and look CGU here. So I have ARG. This is amino acid ARG translated from this sequence. ACC here will be THR. UUA glutamine. So we have now the sequence amino acids attached together. This is giving polypeptide chain, can give us different protein, can give us proline, glycine, phenylalanine, valine, histidine, arginine. All are these amino acids and the polypeptide chain formed from it. So the same as what we translated now. Methionine, arginine, threonine, and leucine. The same here. If you want, I want to give you 2 minutes, try you to use the table, the codon and anticodon table to translate this RNA into the protein. Write the sequence in the chat. And if anyone still don't know how, just keep up with me. I will match this sequence, for example. I will go and look up AUG. Where is AUG? AUG, I will go through each one. AUG, yes, here, I found AUG. AUG, it's matching with what? You look at the name beside AUG, it's MET. MET, it's amino acid. Amino acid is the building unit of protein. New amino acids together, small balls together, amino acids will make a long chain of polypeptide chain or of a protein. So we have translated this. Let's go to ACG. ACG, I need to go and look at the table. ACG. ACG, I will look at A. Here, all the A. Here, we'll start with all the A. So A, ACG, cannot find. ACU, ACG, here, yes. ACG, it's matching with this. Okay? Is THR. Threonine. A is the, okay, no problem, it's LYS. UAA, yes. And yes, Malik, very good. Waiting for the rest. UAA. Stop. Yes, very good. Why stop? To stop translating. It is giving order, signal, stop translating. Now we need this protein. We said that methionine, it will always start the translation process. And also we have stop codons. When you go translating and you will see one of these UAA, UAG, UGA, it will tell you to stop. It will be translated in the table with stop response signal to stop the translation. Okay. For the last activity for today, I want you to try to order this process, protein synthesis, to put them in the right order. I will give you letters. You put them in the right order. A, B, C, D, E, and F. You tell me A, it's number 1, step 2, 3, or what. You put the letters in order according to the right steps. Okay, Nora, yes. Was it? Very good, thank you. Malik, I'm waiting your answer too. I just want to make sure you got the answer before we leave today. So here I will tell you the right order. It will be, okay, this is right, Zuzu. Okay, this is last one, it's telling me last, and this is how it looks like. This is the protein, alanine, glanine, these are where AGU, for example, and we translated it to one amino acid, and the amino acids together will make a polypeptide chain. Exactly. So this is will be number one. This will be number two. Three will leave and go to the cytoplasm. In the cytoplasm, it will go to the ribosome. In the ribosome, the tRNA will find the right amino acid to match it with the mRNA, and the last will forming the polypeptide.